Thanks for that introduction, Fernando. And as Fernando said, my task here is just to give you a bit of an introduction to total body PET technology and its unique capabilities. And I'll just give you a little bit of a taste as well for some of the work that's already been done by others on using this technology and what the opportunities might be for us here in Australia. So for the first couple of slides, if you've heard me give this talk before, this will be a little bit repetitive. I have talked about this quite a lot, but for those who haven't heard this before, when we do a PET scan using a conventional PET scanner, we have a series of detector rings that surround the patient, and typically those detector rings cover an axial field of view here of about 20 to 30 centimeters. And so that's enough to cover and image a single organ at a time. But often we want to image the whole body. And if we want to get an image of the radio tracer distribution in the entire body, we have to move the subject through the detector gantry in much the same way as we would for a CT or an MRI. And that's a very inefficient way to collect the data, remembering that the signal in the case of PET comes from within the patient. So we typically collect less than 1% of the available signal. So it's very inefficient. So total body PET, the rationale is a very straightforward one. The idea is to simply cover as much of the body as possible with radiation detectors or PET detectors. And when you do that, you boost the overall sensitivity and efficiency of the system by at least an order of magnitude. So you get a very sensitive instrument. And in addition to that, you have the possibility now to acquire dynamic data from the entire body. And you can do that at relatively high frame rates as well. If you've got that data, you can then extract the kinetics of the radiopharmaceutical from all the individual organs and tissues of the body. So you can create really quite rich and valuable data sets for research studies. And this slide just shows you a little bit what that looks like, what a dynamic scan looks like. This was one of the first dynamic scans performed by our colleagues at UC Davis. That's a patient who was injected into a vein in the foot with F18 labeled fluorodeoxyglucose. So I'll just run that video. This is a time lapse showing the tracer going through the circulatory system, out through the heart and lungs to the periphery. As a time lapse over 1 hour of this dynamic scan. And you see the tracer gradually accumulating in the peripheral organs and tissues. You see quite intense uptake in the brain, the myocardium of the heart, some uptake in the liver and spleen and in the soft tissues of the body. And you can also see the tracer being excreted through the kidneys into the urinary bladder. This is really quite unique data. It hasn't been possible to acquire this type of dynamic tomographic data of an injected biomolecule in the human body, covering the entire human body. So it's really a unique capability. And the UC Davis team have also shown that there's some information that can be extracted from that early phase of these dynamic scans that we weren't previously able to access. So this study demonstrated that you can acquire data at really quite high frame rates of just 100 milliseconds. And from that data, you can extract information about the perfusion to individual organs and tissues. And then from the later phase of these dynamic scans, you can extract the metabolic information as usual. So I'll just run through some of the unique research capabilities that total body PET provides. So I've talked about the ability to capture the pharmacokinetics of injected biomolecules from all organs. So that means you can investigate disease pathways and novel therapeutic targets. You can do biomarker discovery and validation, and you can do radioligand and drug development. You can also measure signaling between organs. So using receptor binding ligands, we can study the signaling of via neurotransmitters, for example, between the brain and the spine, or between the brain, the spine, and the peripheral muscles in neuromuscular conditions such as motor neuron disease, or between the brain and the gut in the case of Parkinson's disease and diabetes. We can also track imaging probes over long periods of time. So, for example, we might want to study circulating activated macrophages and T cells to study the body's adaptive immune response. We can look at labeled nanoparticles and novel drug delivery systems, and we can study environmental toxins and their distribution through the body. And finally, we can potentially accelerate clinical trials that have an imaging component. So the vast boost in sensitivity of these systems means you get greater statistical power in the imaging data, which means fewer imaging studies to achieve the same surrogate endpoints. I'm just going to show a little bit of the data that's been collected from our colleagues at sites that have this technology, and I'm going to focus initially on infectious diseases just as an example of what this technology can do. So the first couple of examples come from our colleagues at the University of Groningen Medical Center in the Netherlands. And they've done a lot of studies looking at the role of total body PET in studying bacterial infections, particularly in the intensive care context. And this is one of their published studies of a 46-year-old man, a critically ill man in ICU who had multi-resistant staph infection. And you can see from the images that there are multiple abscesses in the chest and the hips, as well as multiple other sites of infection in the lungs and elsewhere. And I just want to point out as well that this image was acquired in just 3 minutes. So this patient was brought down from ICU to the nuclear medicine department and was back in ICU within about 15 to 20 minutes. This second case is a 10-week-old baby. And by the way, this was, the tracer was FDG, and it was also for this baby. This was a 10-week-old baby with sepsis of unknown origin. And once again, it was just a 3-minute scan. The baby was not sedated. It was, so with the short scanning protocol and doing the imaging just after the baby was fed, you could do a quick scan while the patient was asleep. And what the scan revealed was 2 sites of infection. One you can see here in the foot, and also here on the fused image, and another one in the myocardial space. And this site here in the foot was identified as the site of the staph infection, and this site here is a probable site of endocarditis. I should also mention that in this case, the scan was performed with a very low dose, radiation dose, around about 1/10 of the usual injected dose. And this third case here comes from our colleagues at UC Davis. And they've been using a labeled mini body that binds specifically to CD8 positive T cells. And I've been using that together with total body PET to track activated T cells in patients with long COVID. So the image on the left here was acquired just 1 hour after injection of this labeled mini body. And you might be able to notice there seems to be elevated uptake in the bone marrow, and compared with their control group, this was significantly elevated uptake. There's obviously a lot of uptake in the spleen as well. And this image 48 hours later shows the signal having indicating that the T cells have trafficked away from the bone marrow. There's a little less uptake in the bone marrow, and there's lots of uptake in all of the involved lymph nodes, and also in the thymus and tonsils. So a really unique tool for studying the adaptive immune response in this viral infectious disease. And I use these 3 examples here because they do highlight 3 of the features of total body PET. The first one demonstrating the ability to do fast imaging, The middle one, the ability to do low radiation dose imaging, and the third one, the ability to do late, or if you like, delayed imaging, when you're looking at cells and molecules that have very slow transport around the body. Now, all of those examples involve static imaging, but as we mentioned, there's some really rich information in the dynamics as well. And the group from the University of Vienna has developed some really nice open source software tools to extract that dynamic information. Firstly, they use an AI-driven algorithm to automatically segment all of the organs in the total body PET study. And then they've taken some of the connectivity analysis methods that have been developed in neuroimaging and extended them to whole body dynamic analysis. And in this case, they're showing the connectivity maps comparing a group with Infectious with those who have normal muscle metabolism, and looking at the connectivity in relation to the gluteus maximus muscle. And all of that is quite timely, actually, because in parallel with these developments around total body PET, there's a lot happening in the systems biology field as well. And groups who have developed very sophisticated metabolic models at the cellular network and tissue levels are now trying to expand those models to look at metabolic networks throughout the entire body, such as this one. So this is a whole body metabolic model for the male called Harvey, and there's a female version called Harvetta. But the interesting thing about these models is that they can model more than 80,000 biochemical reactions, but and parameterize those reactions, but also that it's able to look at metabolic transactions between organs, and also between the human body and the microbiota in the gut. So some really interesting developments there. I think the opportunity there for total body PET is to help validate these types of whole body metabolic models, and also to generate and test hypotheses. So all of what I've shown you so far is looking at the whole body and systems type approaches. But of course, some people are only interested in imaging single organs. So if you're a neuroscientist, for example, what does total body PET have to offer? Well, there's a number of things. Firstly, you get better image quality. You're going to get much higher signal to noise ratio. You're going to get better spatial resolution. I think Amanda will show you some examples of that. Importantly, you also get better quantitative capability, and I'll show just one slide illustrating that point. Also, of course, the brain is connected to some interesting stuff below the neck, as I've already alluded to. So there's some opportunities there to look at signaling between the brain and other organs. And just like in those other examples, you can also apply the low radiation dose capability to study radiation sensitive populations such as pediatrics. So just to touch on the quantitative capability, so when we do a dynamic brain scan on a conventional PET scanner, we, as I said, we cover enough of the field of view to cover the entire brain. And from that data, we can apply pharmacokinetic models like this one shown on the left here. But if we want to extract the quantitative estimates of these parameters, we need to know what the radiopharmaceutical concentration is in arterial plasma. And to do that, we need to take arterial blood samples. So that's a fairly invasive procedure. And when we do that, that allows us to generate parametric images where each individual voxel represents a quantitative physiological parameter, in this case, the binding potential of that particular receptor binding ligand. But with total body PET, of course, we don't have to take arterial blood samples anymore because we've always got the heart in the field of view. So we can do these types of studies non-invasively. And beyond that, of course, there's the possibility and the opportunity to apply some of the techniques that have been used in the brain to the rest of the body. And this is a paper that literally just came out this morning showing how you might be able to do this with total body PET data, looking at connectivity between different parts of the brain and individual organs. And you can extend that a little further to do network-based statistical analyses in a way that has been done extensively in neuroimaging, but now looking at networks throughout the entire body. So in this talk, I've only had time to really just touch on a couple of examples in neuroscience and infectious diseases, but I really think the opportunity space is enormous. And so the idea will be to take advantage of those unique capabilities of total body PET, together with the Sydney Imaging platform and the NIF network, to exploit those capabilities to look at metabolism, signaling, cell and nanoparticle tracking, therapeutic response, and all of these things in order to address some of the big challenges in the study of complex systemic diseases.